Welcome to today's presentation on the ICH Q14 guideline titled Validation of Analytical Procedures. This is the revised version of Q14, officially adopted in November 2023. It significantly enhances regulatory clarity and modernizes validation principles to include a wider range of analytical techniques, including multivariate and non-traditional methods. Q2 works closely with ICH Q14, which addresses the development of analytical procedures, forming a comprehensive framework that spans the entire analytical lifecycle, from development through validation, routine use, and change management. The scope of ICH Q2 encompasses analytical procedures used for release and stability testing of both drug substances and finished pharmaceutical products. It also applies to methods used as part of the control strategy under ICH Q10. Importantly, Q2 is designed to be phase-appropriate, meaning it can be used from early clinical development to commercial production. This ensures that analytical procedures are validated with the same rigor regardless of development phase, supporting flexible yet scientifically justified implementation. Q2 specifies 6 key performance characteristics that form the backbone of method validation: specificity or selectivity, accuracy, precision, range, detection limit, DL, and quantitation limit, QL. Additionally, robustness is addressed, although primarily during development, as outlined in ICH Q14. These characteristics ensure that analytical procedures are not only suitable for their intended use, but also consistently deliver reliable results under expected conditions. The guideline also recognizes the potential for technology-inherent justification for some characteristics. Specificity or selectivity is the ability of an analytical procedure to measure the analyte of interest without interference from other components such as impurities, degradation products, or matrix constituents. Demonstrating specificity can involve spiking known interferences, applying orthogonal methods like LC and NMR, or using built-in technology features such as spectral resolution in MS. The goal is to ensure that the measured signal truly represents the analyte and is not confounded by related substances or excipients. Accuracy and precision are cornerstones of validation. Accuracy is assessed by comparing test results to known values using reference materials, spiking studies, or orthogonal procedures. Precision is evaluated through repeatability, same analyst and equipment, intermediate precision across days, analysts, or instruments, and reproducibility across laboratories. Q2 also permits combined evaluation approaches using prediction or tolerance intervals, which is particularly useful when working with multivariate or bioanalytical methods. Reportable range refers to the span of analyte concentrations over which the method performs acceptably. Validation must demonstrate accuracy, precision, and response linearity or functional equivalence across this range. For detection and quantitation limits, methods include signal-to-noise ratio analysis, statistical derivation using slope and standard deviation, and visual estimation. The guideline provides flexibility for nonlinear responses, which are common in immunoassays and biological methods. Multivariate analytical procedures, such as those using NIR, Raman, or other spectroscopic methods, rely on multiple variables and often do not use reference standards during routine testing. Q2 defines validation steps for these models, including calibration with known samples, internal cross-validation, and independent validation using external datasets. Performance metrics such as RMSEP, root mean squared error of prediction, and RMSCC, calibration, are essential for assessing model reliability. Lifecycle maintenance, including recalibration, is also emphasized. Validation is no longer a one-time event. It continues across the method's lifecycle. Revalidation may be required following method changes, lab transfers, or new equipment introduction. The extent of revalidation depends on the risk and impact of the change. Q2, R2, supports partial, full, or co-validation strategies. Co-validation across multiple sites can serve both as validation and technology transfer, helping standardize analytical procedures globally across an organization's network. When submitting validation data to regulatory authorities, the data must be well organized and presented in CTD format, specifically in sections 3.2.S.4.3 and 3.2.P.5.3. Submissions should include experimental design, methodology, validation results, and statistical evaluations. Reference to Q14 development data can be used to support the rationale behind the validation design or to justify abbreviated validation using prior knowledge. Transparency and completeness are key to minimizing regulatory queries. To summarize, ICH Q2, R2, is a robust, modernized guideline that accommodates both traditional and advanced analytical techniques. It introduces life-cycle-based validation, supports science and risk-based approaches, and aligns with Q14 to create a unified analytical strategy. By adopting Q2, R2, organizations can ensure analytical procedures are both scientifically sound and globally harmonized, ultimately leading to more efficient development and regulatory review. Thank you for your attention. I encourage you to consider how these guidelines can be incorporated into your organization's analytical validation framework. Feel free to ask questions or share experiences during the discussion. The clarity and flexibility introduced in ICH Q2, R2, offer significant opportunities to streamline validations, reduce regulatory challenges, and maintain high standards of pharmaceutical quality globally.